Welcome everybody to Module 10. This is Mr. Koker, and we will be covering polysaccharides, or long-chain carbohydrates, and also lipids. So this is a bit of biochemistry. As you study this module, keep in mind the different kinds of polysaccharides and the different kinds of lipids, and how their structures can affect their properties. So those would be the kind of things to watch for on the quiz this Friday on Module 10. Also during this module, since the content amount of material is not as much as it was for the organic chapter organic chapters that we just finished, this would be a great week to get some major progress done on that drug project. Get lots and lots of checkpoints done, as many as you can this week, because I think you'll find that the amount of material in Module 10 is not as much as it was in Module 9. We're coming up on Thanksgiving break, and after Thanksgiving break is when that drug project is going to be due. So we want to get some work done on that this week and make some major progress. So without further ado, polysaccharides and lipids. Starting with polysaccharides, poly is a prefix meaning many, and then saccharides are sugars, and so when you sugars of course are in the carbohydrate family of nutrients, and so when you string simple sugar molecules together into longer chains, then you get some polymers called polysaccharides. So these are polymers of these monosaccharides. So we would call a simple sugar like glucose or fructose a monosaccharide. It's in a carbohydrate form, meaning it's made up of chemical formulas that include carbon and then water molecules. Generally, to take the polymers and form them from the simple sugars, you will dehydrate, meaning you will take water out of the chemical formula in order to link the two polymers together. So for instance, sucrose is C12H22O11. It is made up of a combination, a linkage between glucose, C6H12O6, and fructose, an isomer, which also has a formula C6H12O6. So if you take C6H12O6 and add it together with itself, then you get C6H12O6 times 2, you get C12H24O12. But if you take a water away when you form the linkage, you get C12H22O11, which is sucrose. Now that's linking up two monosaccharides, but then you can continue to link them together into hundreds of these monosaccharides in a chain. Polysaccharides are important in both plants and animals. They store energy, so they're a major way of storing energy. They make up the structures for plants, and so stems and leaves and so forth get their strength from the polysaccharide cellulose. They are also important in cellular recognition, so they can appear on the surface of the cell membrane as a way that cells can recognize each other and somewhat communicate between cells. The difference between different kinds of polysaccharides is not so much in the monosaccharides that make them up, but rather how those monosaccharides link together. And so different linkages between these sugars, and also different amounts of branching in the chain of these polysaccharides, can create the different forms of these polysaccharides. So here's an example of how linkage can make a difference. You can see these hexagons here are representing the simple sugars, and if you were to count these up, you'd get basically the formula of a simple sugar here. But C6H12O6, or maybe one less water because of the linkages that you have between them here. But amylose is the name of a polysaccharide found in the starch in potatoes, for instance. So potato starch is amylose. And the structure of the linkage between the simple sugar molecules is pointed out here in the little pink words here, alpha 1,4 glycosidic bond. And so that means basically that the simple sugars are linking up using the 1 carbon on one simple sugar and the 4 carbon on another simple sugar. And so it's a 1,4, and they're calling the linkage there a glycosidic bond. But it's of the alpha variety, and down below you can see the geometry is if the same linkage between the 1 and the 4 are a beta linkage. And so simply the different way of linking for that alpha versus beta can give us the difference between potato and cotton. So cotton is going to be mainly cellulose, and it's made up of beta 1,4 glycosidic bond, creating these cellulose fibers, and that's the difference between cotton and potato. The difference is in the linkage. So some important polysaccharides you should know about are amylose and amylopectin, which are plant starch. So if you have different plants that you eat that contain a lot of starch, potatoes or corn starch or other kinds of plant starches, these are useful in plants for storing energy. So plants are going to take the simple sugars that they make via photosynthesis. Photosynthesis makes glucose, and they're going to store those glucose molecules in various structures, like storing it in the corn seed for the use by the new plant, or storing some plant starch underground in the roots as a potato does. So this is energy storage. Now animals can come along and steal that energy by eating those corn kernels or those potatoes, and it's actually the amylose and amylopectin are both able to be digested by most animals, including us. We can eat potatoes, and we can eat corn, and we can get the energy from them by simply digesting or breaking down those long chain polysaccharides into simple sugars, and then that can give us energy. Glycogen is another form of polysaccharide that animals can generate within their own bodies. So animals can take, if you have, say, an overabundance of sugar in your body, simple sugar molecules, your body can convert those to a type of starch or a type of polysaccharide called glycogen, and store that sugar in your body. And so glycogen is a way that animals store polysaccharides or sugars in a form of a polymer. Cellulose is another form of polysaccharide that's used to form plant structures. The linkage, which is that beta 1,4 glycosidic linkage, the linkage actually prevents animals from being able to digest the cellulose, and so we can't eat tree bark and get any energy from it. I can eat a potato and get energy, but I can't eat a leaf and get energy. I can't eat a tree bark. I can't eat the cellulose material that's found in plant structures. Cellulose is the main ingredient in the cell wall of the plant cell, and the cellulose fibers and the cellulose material gives the stiffness to a plant stem or the structure of a leaf. There are animals that can eat plants, plant structures, like eat grass or eat leaves and digest them, but they do this by having a symbiotic relationship with bacteria in their stomachs. that allows them to break down the cellulose. So all your grazing animals, your horses, your cows, your goats, your animals that can eat grass, or animals that can eat leaves like a giraffe, and break those down into simple sugars, are using bacteria that can break that beta 1,4-glycosidic linkage down and break down the sugars, and then you can those animals then can use those sugars. Now we and many other animals do not have those bacteria in our stomach, and so as a result we don't eat cellulose. Sometimes we do eat cellulose. So you eat a leaf of lettuce, you're eating cellulose, but that cellulose becomes fiber, which then goes through your system and helps your digestive system, but it doesn't give you any energy. You do not retain the sugars in that cellulose. They just become fiber to help you go to the bathroom. So some animals can digest cellulose into simple sugars by using bacteria, but some cannot. Okay, moving on to the second main topic, lipids. Lipids are generally nonpolar molecules. I don't know if you recognized on those sugars that there were a lot of OH groups, a lot of ability to form hydrogen bonds with water and be soluble in water. So sugars are soluble in water, but lipids are not soluble in water because lipids are mainly just hydrocarbons. They'll have minimal parts to their molecules that have oxygen or OH groups and have the ability for hydrogen bonding with water. So lipids are soluble in organic solvents, but not water. Lipids can be made of either fatty acids, which are long chain hydrocarbons with carboxyl or carboxylic acid groups on one end, but the long chain is nonpolar, and then the carboxyl group is polar. And then a second class of lipids are lipids called steroids, which are made up of cyclic hydrocarbons, hydrocarbons with hexagons and pentagons, cyclohydrocarbons, and the steroid molecules have a specific function in the body as well, as we'll see. So this allows us to break lipids down into two major classes: the lipids that have fatty acids, which include the waxes, the triacylglycerols, and the phospholipids, and the lipids that are in the steroid class, which include cholesterol and also the steroid hormones. So this flowchart shows the two classes of lipids. You can see here that the fatty acids are the larger classes of lipids. The basic structure of a fatty acid is a long chain, so we see this zigzag line with a COOH on the end of it in this orange box here called fatty acids. That's a long hydrocarbon chain, lots of carbons and hydrogens, lots of nonpolar portion to the molecule, but it does terminate on the end of the molecule with a carbon double bonded O, single bonded OH, or a carboxyl group. Now in the fatty acids there are three major categories, and they are the waxes, and the waxes are found in some structures like plant structures, and they consist of fatty acid connected with an ester reaction, esterification reaction, to a long chain alcohol. And so you saw in the last organic lab that an alcohol and a carboxyl group can lose water and form a ester connection between the long chain of the fatty acid and then an ester functional group followed by a long chain alcohol. And so you can have two long chains of carbon and hydrogen on each end, the left and the right end, and then in the middle will be a carbon double bonded O and a single bonded O to link the chains together. So those are called waxes. Then you have your triacyl glycerols. Glycerol is a 3-carbon 3-hydroxyl alcohol. So if you think of 3 carbons in a chain, and each carbon in the chain has an OH group, a hydroxyl group. So then that would create 3 positions on that chain of 3 carbons where a fatty acid could use its carboxyl group to react and connect to the 3-carbon chain, the glycerol. By reacting the fatty acid with the alcohol, you can create an ester connection between the glycerol and the fatty acid. And so you end up with this sort of connection here of 3 long tails of fatty acid molecules, and they're all connecting to a 3-carbon chain, which is sort of tying them all together into a 3-carbon chain connection here. And so the connection between the glycerol and the fatty acid is the formation of the ester linkage, much as we talked about before on the waxes. It's an ester linkage between the fatty acid and the glycerol molecule. But it connects 3 different places, and so you have 3 fatty acids all connected to one. You may have heard of triacyl glycerols, also called triglycerides. So think about your triglyceride levels in your blood. They're talking about the content of this molecule, triacyl glycerols, in your blood, and so it's a type of fat basically that can be found in your bloodstream. And if it's too high levels, then they're gonna try to change your diet to before you have a heart attack. Okay, and then something very similar to the triacyl glycerols, we have our glycerophospholipids. Here we have once again the glycerol molecule, which is a 3-carbon alcohol, but then it links up to 2 fatty acids, but the 3rd linkage is a phosphate group and then an amino alcohol. So the fatty acid part is going to be nonpolar, but the phosphate amino alcohol part is going to be polar. And so this gives us a molecule which has both a polar region, the phosphate amino alcohol region, and a nonpolar region, the fatty acid region. And we'll find later that these glycerophospholipids are important in the structure of the cell membrane. The second class of lipids are the steroids. So you see the basic structure of a steroid is this 3 hexagons of carbon. So you have the 3 hexagons, and then on the right side there you see the pentagon of carbons. And so it's these 4 cyclohydrocarbon, hexagon, 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 and pentagon that have this very unique structure, which make up the steroids. And the 2 main kinds of steroids are cholesterol and then the steroid hormones. Okay, let's take a look at what we mean by a fatty acid. Fatty acids are long-chain carboxylic acids. By that they mean we have lots of carbons, 12 to 18 carbon atoms in a chain. And then on the end there's a carboxyl group, a C double bonded O, OH. Now because there are so many carbons and hydrogens in the long chain, these are going to be insoluble in water despite the polar end, which is the carboxyl group. There's so much nonpolar in the molecule that they're insoluble in water. So you find that many of the fats and oils and lipids that we have in our cooking, for example, the olive oil, the bacon fat and all that, they're very hard to clean up because of the insolubility in water. You got to get some Dawn dishwashing liquid if you're going to get that to wash away with water. Fatty acid chains can be saturated or unsaturated. Now remember back in organic chemistry, the last chapter, last module, that saturated means all the carbon chains are single bonds, versus unsaturated would mean that there would be some double bonds in there. This can make a difference in the properties and also the healthiness of the fatty acids. healthiness in meaning when you eat them. How's it going to act within your body? So to show the difference between a fatty and a fat that's saturated versus an unsaturated fat, we see here palmitic and palmitoleic acid. And in palmitic acid you see the zigzag line there. That's just the skeletal structure of a long chain hydrocarbon chain, and then it terminates on the end with a carbon double bonded OH, a carboxyl group. Notice in the palmitic acid all of the carbon-carbon bonds are single. Contrast that with the middle of the chain for palmitoleic acid. You have a double bond there. And so the double bond means that that's an unsaturated fat or an unsaturated fatty acid. So saturated is all single bonds. Unsaturated would be one or more double carbon-carbon double bonds. If you have more than one, they would call it polyunsaturated fats. And so you may have heard that, unsaturated fats, polyunsaturated fats. These are the healthier ones, as we'll see. The problem with saturated fats is when they contain only carbon-carbon single bonds, the flexibility of those long chain hydrocarbons allow them to fit closely together. They can mold themselves to the shape of the neighboring fatty acid molecules, and because they can get a lot of closeness and they can pack closely together, it will cause stronger attractions between those chains. And so saturated fats tend to be solid at body temperature or room temperature, and we see that with bacon fat and butter fat, that the saturated fatty acids are actually going to be solid because they have stronger attractions. Now you don't want fats in your body, particularly in the bloodstream, fats in the bloodstream, to be in the solid state. Those are going to be clogging the arteries, and so we want them in a liquid state if it's going to be healthier when it moves through the bloodstream. So with high melting points, these are going to be solids at room temperature and pretty much at body temperature as well. Now when you have oils, which is what we call the unsaturated fatty acids, or molecules containing the unsaturated fatty acids, are going to be oils instead of fats. Fats generally denote solid, oils denote liquids. So they have very similar structures, long chain fatty acids, but the double bonds, which can be either cis or trans, right? Cis fats and trans fats. The double bonds are going to mean that there's a kink in the chain, and that kink in the chain is not flexible. A double bond can't rotate like a single bond can, and so there's less flexibility. With less flexibility, it becomes more difficult for these chains to line up side by side. The kink in the chain means the chain is going to have some V shape to it, and then those V-shaped portions aren't going to be able to line up side by side, and as a result your oils are going to be more liquid. So unsaturated fatty acids contain one or more cis double bonds, and these are going to be kinks in the fatty acid chains, keeping them from packing closely. Fewer attractions between the chains means these are going to have low melting points and be liquids at room temperature. And so your corn oil, your olive oil, these have very similar structures to bacon fat and butter fat, but bacon and butter are solid at room temperature because they have saturated fatty acids. The oils are going to be unsaturated fatty acids. You might notice that the saturated fats tend to come from animal sources, like butter comes from dairy, and bacon fat comes from a pig, whereas the oils tend to be more liquid, and they also come from plants. So corn oil and olive oil, these come from plants, not animals. Okay, here's the glycerophospholipid. These are going to be the most abundant lipids found in your cell membranes. You're going to have your glycerol, which remember is your 3-carbon alcohol. That's going to link up with 2 fatty acids and a phosphate and amino alcohol. So you have the glycerol, the 2 fatty acids, the phosphate, and the amino alcohol. The phosphate and amino alcohol part are going to be polar. The fatty acid part is going to be nonpolar. So you're going to have polar and nonpolar portions. So when you have molecules that are both polar on one part of the molecule and nonpolar on another part, this is called amphipathic. So amphi usually goes along with being both. Think of an amphibian, can live on land or in the water. Amphi is a prefix, it means kind of both. So amphipathic means both polar and nonpolar. So here we see sort of a more specific version of what the glycerophospholipid would look like. In the light blue in the center, you see the 3-carbon chain. That's the glycerol. You see the phosphate, the PO4, and then the amino alcohol, which is in the green box there. That's the phosphate and amino alcohol make up the polar head of this molecule. And then in the yellow, we see the 2 fatty acids. Note in this particular picture that the top fatty acid is saturated, but the second fatty acid is actually unsaturated. See the kink? It's a cis connection there, the unsaturated fatty acid. And so we have that 2 nonpolar tails. And so you have the nonpolar tails and the polar head, meaning this glycerophospholipid is going to be amphipathic. Okay, the second class of lipids are called steroids, and so they have this classic structure of the 3 hexagons and the pentagon. This happens to be a picture of cholesterol, which is the most abundant of the steroid molecules in the body, and in some people more abundant than others. Some people have problems with high cholesterol. We have methyl CH3 side groups. We have alkyl chains, so that would be those zigzag carbon chains that are coming off of the pentagon there. And also you're going to have the OH that's going to be attached on that steroid nucleus. So in the lower left hand corner you see the OH group. Hence the suffix "-ol", cholesterol, meaning that there's an alcohol group on it, the hydroxyl group, the OH group. So think back to your organic chemistry. This is some extra practice on these organic structures. We're not done with organic chemistry, even though we're doing biochemistry. We're still not done with organic chemistry because it's the main chemistry behind biochemistry. So it's cholesterol, OH group on the steroid here. So you can tell by looking at this, lots of carbon, lots of hydrogen. This is a very nonpolar molecule. The only polarity it has, the only ability to form hydrogen bonds with water would be the OH group, and that is so much outnumbered by the hydrocarbon cyclo structures and the alkyl chains and the CH3 groups. So the problem with cholesterol is cholesterol clogs arteries. Here's a couple of cross sections of a normal open artery and then an artery that's clogged by cholesterol plaque. So we get cholesterol when we eat various meats, milk, and eggs, generally from animal sources. But if you just were total vegetarian, you would still have cholesterol in your body because cholesterol is synthesized in your liver. And so it's not like we need no cholesterol. That would actually be unhealthy. We need some cholesterol in our bodies. It's just when it gets too high that it becomes a problem and starts to clog the arteries. So we need it in order to make cell membranes for brain and nerve tissue to make steroid hormones, and also in the production of vitamin D. So cholesterol needs to be at a certain level in our blood and in our bodies, just not too much. But many of us like to eat that meat and milk and eggs, and that tends to raise our cholesterol levels. When the cholesterol gets too high, it tends to form clogging in the arteries by the production of plaque, or basic solid material that collects on the walls of the arteries and slowly fills in the arteries over time and blocks the blood flow in the arteries. Now, if that happens in an artery that feeds the heart, then you can have a heart attack, right? So looking at some foods that have very high cholesterol content. So let's say that your doctor told you that you needed to cut down your cholesterol levels. What would be some foods you might want to cut out of your diet? Liver. Liver is where the body makes cholesterol, and so the liver, if you eat beef liver or something like that, or chicken liver, you're going to have high cholesterol in that liver, so you're going to be putting a lot of cholesterol in your body if you eat liver. Many people don't like liver. I happen to like liver. I like the taste of liver, chicken livers, beef liver, cooked with onion or bacon, but it does have a lot of milligrams of cholesterol. Eggs are high in cholesterol. Lobster, fried chicken, hamburgers. So these would be cholesterol. I don't see cheese on here, but I know cheese is something that some people avoid if they're trying to cut their cholesterol. Margarine, which is actually made from plant oils, it's a replacement for butter. Margarine has no cholesterol. It has fats, but it doesn't have cholesterol in it. So if you go to the doctor and the doctor says looking at your blood plasma and testing your blood plasma, that your cholesterol levels are above 200 milligrams per deciliter, then you're going to be told that you need to get your cholesterol levels down. And so they'll tell you to change your diet, and changing your diet doesn't work. Maybe they'll put you on cholesterol medicine. But they're trying to avoid the the plaque that makes you have a heart attack, right? The movement of these lipids through the bloodstream is typically in the form of lipoproteins. The problem with getting lipids from our diet to our cells is that lipids are nonpolar, and the blood that takes nutrients to the cells, of course, is water-based and is polar, and so lipids do not dissolve well in our blood. So as a result, our body will package the lipids in these structures called lipoproteins, which will allow the lipids to be transported through the bloodstream. So what happens is the lipids are typically packed in the center of these lipoproteins in a nonpolar region, but then a surface of glycerophospholipids. Remember, glycerophospholipids have nonpolar tails, which will point in towards the nonpolar lipids in the middle there, and attach to those nonpolar lipids, and then a polar head, and also using certain membrane proteins to hold everything together, because proteins are typically molecules used for structure. So by gluing them together with these membrane proteins, and then having these glycerophospholipids on the surface, which is very similar to a cell membrane on the surface, you can create these little packets of lipids that can then be transported through the bloodstream, because the polar heads of the glycerophospholipids can interact with water, and so the blood is going to see this packet coming through with a polar surface and interact with that polar surface, whereas hidden inside that packet is a bunch of nonpolar lipid molecules. So it's combining lipids with proteins and also phospholipids like glycerophospholipids. and it makes them soluble in water because the surface is going to have these polar glycerophospholipids on them. Now, the lipoproteins are a variety of types that differ in density and composition and function. We're not going to go into detail on them, but mainly you go from your lowest density, which would be the chylomicrons, and the very low density lipoproteins, and then the low density lipoproteins, and finally the high density lipoproteins. Notice that the low density lipoproteins are mostly triacylglycerols, which are your triglycerides, whereas your high density lipoproteins are mostly phospholipids, like the ones that can be used in cell membranes. And so high density lipoproteins are actually considered healthier because they're mostly protein and phospholipids, as opposed to the low density lipoproteins, which have higher amounts of triacylglycerides in them, and cholesterol esters and so forth. Back to the steroids here. A second kind of steroid besides cholesterol are the steroid hormones. These are going to be chemical messengers that are going to communicate with cells and induce the development of certain body structures. Androgens in males include testosterone, and so it will tell the body to develop male features. And then estrogens, such as estradiol in females, is another kind of steroid that has the classic steroid structure. And of course it's going to have female traits developed based on the chemical messenger telling the cells to develop certain properties. Anabolic steroids are derivatives of these steroids that are sometimes used illegally by athletes to try to increase their muscle mass or speed healing and so forth. So most sports are going to outlaw these the use of anabolic steroids because you get an unnatural advantage by using these anabolic steroids. Also because they're trying to protect their athletes from having some of the major side effects that can happen here, including fluid retention, hair growth. For example, women that use steroids for athletic might have hair growth all over their bodies or something, sleep disturbance, liver damage, and there have been a number of athletes that have died from the side effects of taking large quantities of anabolic steroids, and so it can be a very dangerous thing to do. Now, there are a couple of things in the chapters on carbohydrates and lipids that you can skip over. And that would be anything that discusses the Fisher projections and the difference between reducing sugars and non-reducing sugars. So you do not have to worry about that in the book. Finally, let's talk about cell membranes, because cell membranes kind of tie this all together. Cell membranes are going to contain polysaccharides, and they're also going to contain lipids, including both steroids and phospholipids and things that contain fatty acids. So cell membranes, the purpose of a cell membrane in the body is to separate what's in the cell from what's outside the cell. So to separate the cellular contents from the external environment. A cell membrane is going to consist of a lipid bilayer, so it's a double layer of lipids, 2 rows of phospholipids. So these glycerophospholipids that we talked about as one of the major types of fatty acid based lipids. These glycerophospholipids, which have polar and nonpolar regions, or amphipathic, are going to make up most of the cell membrane, and they're going to be a double layer in the cell membrane. And what they're going to do is they're going to have the polar head facing outward into the external environment, because outside of your body's cells is a watery environment. The interstitial fluids between the cells is water, water-based. And so you want the surface of the cell, much like those lipoproteins we saw, to be polar. And so you're going to use the polar head on the outer surface of the cell, and then inward toward the middle of the cell you're going to have the nonpolar tails. But since the inside of the cell is also water-based, inside your cell you have water and things dissolved in water, you're going to have a second layer, which is going to take its nonpolar tails and point towards the nonpolar tails of the first layer. And by doing so, then point their polar heads toward the inner surface inside the cell. So this is going to form a type of sandwich with the polar heads making up both slices of bread, and then the inside being of the sandwich, whatever's in the sandwich between the bread, is going to be the nonpolar tails from both layers. This is called the fluid mosaic model and of the cell membrane. And not only does it contain these glycerophospholipid bilayers, it also contains proteins, which are going to help with the structure of the cell membrane and also specialized regions of the cell membrane. Carbohydrates. One of the things carbohydrates do is they help with cellular recognition on the surface of the cell. And also cholesterol, which can affect the fluidity, or the liquid flexibility, I guess would be a better word for it, flexibility of the cell membrane. It's going to have unsaturated fatty acids in the cell membrane. Unsaturated fatty acids are going to make the cell membrane more fluid-like rather than rigid. Now this has a function. In colder regions, say a reindeer or a Arctic wolf would actually contain higher amounts of unsaturated fatty acids in their cell membranes because of the cold temperatures, which tend to solidify liquids. Liquids tend to freeze when they get cold. Because of that tendency, the cell membrane needs to stay fluid, needs to stay flexible. And so to keep it more fluid, you would incorporate more unsaturated fatty acids into the cell. And so the Arctic wolf, or something that lives in the very cold temperatures, might have higher degrees of unsaturated fatty acids in their cell membranes. Whereas something that lives in a very hot environment, might, say a desert mouse or something, might actually have more saturated fatty acids, and then those the high temperatures would make those saturated fatty acids fluid. So to keep a degree of fluidity, but not too fluid, but not too solid in the cell membrane, the cell can actually balance that out by the amount of unsaturated or saturated fatty acids, the degree of unsaturated and saturated fatty acids that are included in the cell membrane. So there's some ways of engineering the cell membrane that way. Also proteins, carbohydrates on the surface that are going to be communicating with hormones, neurotransmitters, cellular recognition. Cells are not on an island. Cells work together in the body, and cells have to communicate with each other, and so there are things on the surface of the cell like proteins and carbohydrates that are going to be the way in which cells recognize each other and basically talk to each other in a type of way. So this sort of ties it all together. This is the cell. The nucleus of the cell, of course, is where the DNA and the RNA is produced, and the RNA travels out of the nucleus into the cytoplasm for protein. But that's all in our last module, Module 12. We'll learn about DNA and RNA, but that's in the nucleus of the cell. Then outside the nucleus we have the region of the cell called the cytoplasm, where many of the organelles, the Golgi bodies, the mitochondria, the ribosomes, and so forth are found. But then you have the surface of the cell. The surface of the cell is a very highly specialized structure. It's mainly made up of glycerophospholipids. So if we start in the upper left-hand corner at this blown-up picture of the cell membrane, we have our glycerophospholipid bilayer. So notice it's a double layer. The green spheres are representing the polar heads, and so you have polar heads on the outside surface of the cell, and you also have polar heads on the inside surface of the cell. That would be the hydrophilic region as labeled there. Hydrophilic means water-loving. In other words, that's the polar part, that's the part that can interact with water. And because outside the cell and inside the cell there's water, you want the outer surface and the inner surface of this membrane to be able to interact with water. However, you also have the two nonpolar fatty acid tails on each of these glycerophospholipids. And those tails are nonpolar. And so inside the sandwich of the polar heads, we see the nonpolar tails occupying that region inside the sandwich. And so the nonpolar part can interact with each other. The nonpolar tails can stick together because those are both nonpolar, and like dissolves like. So that would be called the hydrophobic region. And so the cell membrane has on its inside and outside surfaces hydrophilic parts, and then on its inside region there you've got hydrophobic region on the inside of the sandwich that makes up the plasma membrane of the fluid mosaic model. We would call this the fluid mosaic model. Now, incorporated in with these glycerophospholipids are cholesterols, and so those orange spheres represent an interspersing of cholesterol, and the level of cholesterol in the cell membrane can affect its fluidity, as well as the incorporation of unsaturated fatty acids in the cell membrane as well. These dark blue structures actually represent various proteins that are used sort of to hold things together. They're used for structurally. They can also be used as like channels and so forth, ways of getting things in and out of the cell and so forth. So these, and they can be used for communication as well. As we see on the surface, we see some glycolipids and some glycoproteins, which are going to include both carbohydrate side chains and protein surfaces. Remember, proteins and carbohydrates are the main way that cells communicate. So that kind of wraps up our little section here on lipids and carbohydrates, or long chain carbohydrates. Thank you very much.